Recording is started.
All right, everyone. So today we're going to be talking about ATPase, which is a specific type of enzyme that regulates and deals with ATP in the body. So it's a very important enzyme, and that's why we're devoting an entire lecture to it. What we're going to cover in this lecture are first the structure, what the parts of an ATP enzyme are, the function, what it does and why it needs to do that. 3, what the specific reaction in which the ATPase is used, and of course end off with the review. So let's get started. So starting off with the structure, we need to look at the different parts of the enzyme, but something that you need to know in general is that for an enzyme, the function is based on the structure. So the reason an enzyme is shaped the way it is because that shape allows it to do a certain function. For example, a Lego block is shaped a certain way because it serves that function. If it's shaped in a different way, then it serves a different function. So this is what we call this blue part in the middle. It looks like 2 layers with a bunch of spheres on 2 ends and then lines in the middle. This is the lipid bilayer that you find in a cell. And what this signifies is that one side is the inside of the cell, which is the top part, and the other side is the outside of the cell. So if you recall correctly from our previous lessons, you know that there's a cell membrane, and that membrane is this lipid bilayer that's found in light blue. So the bottom is the outside, the top is the inside in this case. And the inside is what we call the F1 region for the ATPase enzyme. This is what we call hydrophilic. So if you break down the etymology of this word, hydro means water, philic means like attracts or to like. So this F1 region actually attracts water. Why is that important? Because water is involved in the reaction. The function of ATPase is that it synthesizes ATP, and other types of ATPase hydrolyze ATP in this region. So hydrolyze just means to use water in a reaction. This ATPase is known as a catalyst. So technically, this reaction that synthesizes ATP could theoretically happen spontaneously. However, it uses a catalyst because of what's called activation energy. That's something that you'll learn in chemistry. And what that is that you need a certain threshold. Actually, no, you need a certain amount of energy in order to start a reaction. And what a catalyst does is that it takes the reaction in a different pathway so that it uses less of an activation energy and therefore is more likely to happen and occurs faster. That's just a purpose of a catalyst, and that's what this F1 region does. It also has 5 functional subunits, as you can see in this diagram, but we're not going to go through that just yet. Oh, looks like I was signed out of my Google Drive. Just give me 1 second. Sorry about that. My apologies. All right, we can move on now. So speaking of those 5 functional subunits, can you move the slide, please? Yeah. So as you can see, there's alpha and beta. There's 3 of each of these subunits, and they create what is called a hexamer. And why do we call it a hexamer? Because there's 6 and they're arranged in a circle. They're what's called catalytically inactive, but it binds to ADP. Now, this is a good spot to discuss what ATP and ADP stands for. ATP stands for adenosine triphosphate, and ADP stands for adenosine diphosphate. What does that mean? They have the adenosine group, but attached to them on the ATP, there are 3 phosphate groups. For the ADP, there's 2 phosphate groups. And how do you make an ATP? You add a phosphate group into the diphosphate to make it a triphosphate. There's also a section called the gamma section. This allows the beta subunit to go through closed open and half open states. What this does is just moves the shape of the enzyme in order to do different things and create a catalytically faster reaction. There's also a delta subunit. It holds the F1 subunit stationary against movement of the axle. So what you want to notice here is that it's actually kind of like a little rotor that turns. And that's the structure of the ATPase. And if a rotor turns, you want to make sure one part of it stays grounded to one side so that it doesn't spin that part as well off. Because if that happens, then it could just like completely mess up the entire process, and it might not change its shape accordingly as efficiently. So there's a delta subunit that holds it stationary. And finally, there's something called the epsilon subunit, and this can actually inhibit the ATPase's activity. What this means is that if this epsilon subunit is activated, then it kind of puts the brakes on this enzyme and stops it from doing that rotation so that it pumps out ATP. This epsilon also aids in proton transport in chloroplasts and bacteria mainly. This proton transport is kind of like the energy, if you may, that drives this motor. Next, we move on to the F0 region. The F0 region, which is kind of located on the inside of the lipid bilayer, is water insoluble, also known as hydrophobic. If you recall F1 as hydrophilic, which means it likes water, there's water inside cells, so that's why the hydrophilic end, the F1, stays on the inside of the cell. However, the F0 region is hydrophobic, so water insoluble. It doesn't like to be around water, and therefore it's going to be on the inside of this lipid bilayer. So if you remember, the tails of each of these lipids is also hydrophobic, so they're going to try to be away from water. So the F0 region is going to be in a spot around those areas. And remember when I mentioned that proton transport? Well, in the F0 region, there's a proton pore in the mitochondrial membrane that lets protons across. It also has 3 main subunits and 6 extra in humans. That's something interesting to know. There's also something that's called protonation and deprotonation, which causes the axle on the inside, if you see this pink arrow, that causes the axle to spin and change out the alpha and the beta subunits. So each time those change, then an ATP is produced. And of course, this happens in the mitochondria because, as we all know, that meme, mitochondria is the powerhouse of the cell. That's where ATP is produced. So this is the inside and the outside of the mitochondria. Moving on, we'll look specifically at the F0 subunits. So look at these specific sections where it's labeled A, B, and C, and that corresponds to what's described on the left. So in part A, it connects the B and the C subunits together. And it also allows hydrogen ions to enter the C subunit. So as you can see in the C subunit, there's kind of like a hole that allows the protons to enter. Part B is a stalk that reaches up to the alpha beta hexamer and regulates function. So if you recall, in the original drawing, there's a hexamer of alpha and beta, and that reaches up to there in order to regulate function. So actually, in this case, it's like the same diagram that I showed you before, but it's upside down. So like the outside, the top is now the outside, and the bottom is now the inside of the mitochondria. And then finally, part C, it's like one of the most important parts. There are actually 6 quote unquote C subunits that make up the rotor ring. These turn and allow the protons to go in and are connected to the hexamer on top with the alpha and beta subunits of F1. This allows the entire thing to rotate. All right, next up I think is Richard, who's going to talk about the function, which is very closely related to the structure.
So the second part of the slideshow is talking about the function of ATPase. You can go to the next slide. So ATPase synthase is a catalytic enzyme, and a catalytic enzyme, well, enzymes are always catalytic, as in they fuel and they allow reactions to progress. And what happens is that it creates ATP from ADP, or adenosine diphosphate, and inorganic phosphate. Now what happens, of course, as we've already talked about, it is found in the membrane of the mitochondrial matrix, and this is F-ATPase specifically. And there's other types of ATPase, there's V-ATPase, and there's also P-type ATPase, but we're talking about F-type ATPase, and this is specifically in the mitochondrial matrix. And these other types of ATPase are found in different parts of the cell, depending if you're a plant cell or animal cell, or just different regions of the cell. So F-ATPase specifically is found in the mitochondrial matrix membrane. And the reason why this particular form of ATPase is important is that it is part of the hydrogen ion gradient created by what we call the electron transport chain. And this connects back to cellular respiration, which is the process, the biochemical process in our cells that allows our cells to gain energy. And what happens is that the electron transport chain essentially moves electrons across the mitochondrial matrix membrane, and the moving of electrons allows for energy to be released, essentially pushing the H plus ions out of the inside of the mitochondria to the outside of mitochondria. Now what happens is that the H plus ions, through passive diffusion, to go down through the membrane because of the difference in the concentrations, it passes through ATPase. And the movement of ATPase eventually causes the production of ATP from ADP and inorganic phosphate. So you could also call this mitochondrial hydrogen ion ATPase. Now, V-ATPase is found in a different part of the cell. It's actually found in the lysosomal membrane, and it is still and we still use hydrogen ions, but it is more hydrogen ions, but also you could also even use potassium ions and sodium ions as well. And this provides the energy specifically for processes that involve the breaking down or lysing, and that's the whole function of lysosomes, to break down particular materials inside a lysosome. So the VATPase provides the energy for those particular functions. So there is a slight difference in the F-ATPase and the V-ATPase. The biggest difference that you should really know is that the F-ATPase rotates clockwise while the V-ATPase rotates counterclockwise. And this is mainly due to not only the kind of the structure. I mean, the structure is generally similar. I mean, the main difference is that V-ATPase contains more parts and more subunits because of its difference compared to the F ATPase, but the main difference is the counterclockwise movement. Now, the other main difference is the movement of hydrogen ions. So what happens is that F ATPase, the hydrogen ions move from the outside of the mitochondrial matrix to the inside of the mitochondrial matrix, while the H plus ions move from the inside of the lysosome to the outside of the lysosome. So they move in opposite directions. So that's the other main difference. Okay. I think we kind of, okay, great. So P ATPase. Now this is found in the sarcoplasmic reticulum. And we actually talked about this the last lesson, not last week. And the sarcoplasmic reticulum is similar to the smooth ER, but the sarcoplasmic reticulum is specifically found in the muscle cells because muscle cells need actually a larger, they need a larger amount of calcium 2+ ions because they need those ions to be able to contract and relax the muscles. So the sarcoplasmic reticulum has this ATPase, also known as calcium 2+ ATPase. And it is also found in the plasma membrane, but mainly found in the sarcoplasmic reticulum. And what happens is that through the movement of sodium and potassium, we are able to then produce calcium 2+, which is the main ion used in muscle cells to contract and relax muscles. You can move on to the next part.
All right, so now we're going to talk about the reaction of ATPase. So what happens when it's actually used? So these are numbered steps, but they're not really occurring in a specific order because this is happening all at the same time and, very fast. So we'll talk about these in this order, but just know that it's not exactly in that order. So first you have to have a hydrogen ion gradient. And once that hydrogen ion gradient is created, then you can have that go through the hydrogen ions, move from that high concentration to the low concentration through this hydrogen ion pump. And so with that movement of the hydrogen ions, you're going to have the ADP and inorganic phosphate bind to the active site, which is going to be that beta subunit. And then you're going to have that beta conformational change, which will actually put the ADP and inorganic phosphate together to make ATP. You can so talking about step 1, so you're going to have the hydrogen ion gradient, meaning that there's more hydrogen ions inside of the inner mitochondrial membrane than outside of the inner mitochondrial membrane. And just for you to visualize the mitochondrial membranes, the mitochondria actually has 2 membranes. So it has that outer membrane, which is, you usually see it in pictures as just like this oval shape. But then the inner mitochondrial membrane is going to have like lots of different turns in it. So with those turns, it gives it actually more surface area than the outer mitochondrial membrane. And with this greater surface area, it makes more room for these this thing called an electron transport chain, which is what creates the hydrogen ion gradient that we need in within the inner mitochondrial membrane versus the outer mitochondrial membrane. So this isn't actually the ATPase enzymes aren't actually outside or going outside of the mitochondria, but they are in the mitochondria at all times. It's just which part of it. So the F0 domain is going to be inside of the inner mitochondrial membrane, and then the F1 region is going to be outside of the inner mitochondrial membrane. So in between the inner and outer mitochondrial membranes. And keep in mind, this is just for F-ATPase. It's not going to be for the V-ATPase or P-ATPase. So first off, you're going to have the hydrogen ions, which we also refer to as protons, just because if you look at a hydrogen ion, it's not going to have that electron there, so it's going to have a positive charge. And so it's basically just going to be a proton. And it doesn't need a neutron there either. So you're going to have this hydrogen ion, and first off, it passes through subunit A. So subunit A, we don't know the exact structure of, but the hydrogen ion has a place to go through, and it's going to attach to the C subunit. And with this hydrogen ion going through and attaching to the C subunit, that's going to cause it to rotate, sort of like a water wheel with the flow of water. It's going to move in a specific direction, and it's going to get through, or using, after 3 hydrogen ions, there's going to be enough energy of rotation to be able to have the combinational change of that beta subunit, but we're not quite there yet. So it binds, the hydrogen ion will bind to the C subunit and cause the rotation of not only that C subunit, but also the that gamma and epsilon subunits, which then connect to the alpha and the beta subunits and cause the conformational change there. So if you note on the side here, you have the membrane domain, the stalk, and the catalytic hexamer, and these are going to be important parts movement-wise. So the catalytic hexamer does not move, and then the stalk and the membrane domain, those are the moving parts. So while the hydrogen ion transfer is going on, and while they're being like taken in and released, you're going to have an ADP and inorganic phosphate molecule bound to the beta subunit in this F1 region. And so as the hydrogen ion moves from the intermembranous space into the matrix, it's going to allow for bonds to form between ADP and inorganic phosphate. And this is a really complicated thing that we're not going to get into, this creation of this bond and how exactly the beta conformational change occurs. But you just need to know that it is created through this use of kinetic energy to mechanical energy. So we're using this kinetic energy that the hydrogen ions have and turning it into mechanical energy with the movement of with the mechanical movement or physical movement of this F0 region, the C subunit, and the stalk of the ATPase enzyme. So with this movement, there's enough energy built up to actually cause the conformational change in the beta subunit, which allows the ADP and inorganic phosphate molecules to bind together into ATP. And just notice one more time, it takes 3 hydrogen ions, also known as protons, to produce 1 ATP molecule. And that's just important when you're looking at the actual reactions. And then I'll hand this off to Adam for the questions.
Yep. All right, you guys can hear me? I guess. Hope so. All right, next slide. Review. What does ATP stand for? Adenine triphosphate. What is the function of F-ATPase? To create ATP from ADP and an organic phosphate from a hydrogen electron gradient. What is the function of V-ATPase? To create ADP and an organic phosphate from ATP using a hydrogen ion gradient in the lysosomal membrane or a hydrogen ion potassium ion gradient in the plasma membrane. How many hydrogen ions does it take to synthesize one ATP molecule? 3 hydrogen ions. What are the 2 regions of ATPase and where are they located in/on the membrane? The F1 region is found outside the membrane, whereas the F0 region is found inside the membrane. What 2 subunits make up the rotating axle? Gamma and epsilon. All right, thank you guys.